wrote to Trixie and I said, 8 hours is enough.
There's nothing you have to do with those.
Good. She thought otherwise.
I was thinking about it back after that.
Let's just say I do it.
I figured, I said, I don't have time to do this.
Yeah, I was yeah.
They torture us enough.
No, it's looking bleak. Although it's just giving me a lot more time on the weekend.
We have
After 8 hours, she finally gave me one piece of paper.
last
year. How you signed it.
Once you get out on the actual probably you just show you amazing pointers about seeing the number of boxes. It's pretty easy once you get there. It's similar to you guys.
The screen is more complex. At least the one I was working with.
There's a way that, yeah, depends on your preference.
He's just shaking his head, thinking,
no way I'm going to do anything like that. I retire.
I was going to ask Werner Sampson in the 80s and Marvin Turk in the late 70s. Did they give you guys a pass? Or did, they have to sit through this training. I can't believe they would put guys that senior through this.
It is. It's a lot. I've been on it a year and I'm still learning the nuances of it. But as far as attending, it should not be.
You guys just have the cameras.
Should not be hard.
Created monster. It's great for documentation. It's terrible to review.
Did you see the why? It's not that great. Well, if you do a good job, it's no better than the person who put the notice.
I can't stand the fact that people push a button and just repeat everything each time. That drives me crazy because I review legal stuff. One page after the other, it's the same stuff. You've got to find one little tiny area that's different. Instead of having a chart this thick and
Bureaucrats think that it's a good chart because there's a lot of words on the page. Drive you nuts.
That's why I like Dragon. I use it the same way you do.
Yeah.
Most of mine are free text.
That's the way they track it. You free text, they can't track the demographics. So there was an article that just came yesterday in the Washington State Bulletin about scribes, and it's a great industry, it's a school just to keep people to be
Starts if I had to, that's what
the only way.
When you go to Israel,
You want to use your
March.
that's not that far. I might play hook. No, you don't. First of all, take out several weeks here. And then the other thing is, you got to ski at my age, use or lose it. They said I could, so I'm looking at that.
Plus, you're not missing any good skiing nights. You're not about that.
Oh, I'm only at the airport.
Stevens is pretty much down to granite in a couple of spots. So the Grimmers can't make granite turn into snow. Have you been to Whistler?
It's not impossible.
Whistler is the top is relatively well covered. I mean, there's still a lot of rocks sticking out, but they've had to make snow to get you to the village. So there's only one way into the village on either side. Better than
What? I don't know.
Anybody need another copy of this, or a copy of this directory, a couple of extras?
Vanderbilt.
Our new administrator. If you ever need to see her, just no, she's been working on it. So she does your first one, 13th. Save the day. let's just save it to the desktop so the transitions are.
So the word of the day is Vanderbilt.
Is that where you trained?
Yes.
Yeah. What city is that in? A nice place? All right. Troy just told me he actually went to Nashville trying to be a country western singer, but that career didn't work out. Just a couple of quick announcements. As I mentioned, there are some more of the directories if you need them, although we mailed them out, so most everybody should have one. Again, the word of the day is Vanderbilt. I'll just remind you, everybody who's listening to me, at the meeting this year, Sunday we'll have a UW Northwest alumni dinner, not sponsored by Pharma. So if you haven't RSVP'd and you want to go, it should be very enjoyable. I've got a restaurant reserved, much like I did in San Diego, and closed the whole place for us for the evening. If you're a fellow or the whole meal will be paid for, your spouse, significant other, and kids, so bring everybody that you want. But if you're any more senior than that, you have to pay the bill yourself. So today, Troy will give us the annual update on immunodeficiency, especially important for old people who don't know any of this and then forget it as soon as he teaches it to us.
Thank you very much. It's a pleasure to be back. And today I wanted to talk about, I think, some interesting new findings in the last year. There's some of these go back beyond the past year, but I think I'm going to try and I'll give an update on some things I think that are interesting that have come to light, really related to immune dysregulation and autoimmunity, and we'll talk about several genetic defects that I think provide some interesting ideas about some critical pathways in autoimmunity. I love this graphic. I pulled this offline. This is the usual regulatory T cell flow cytometry that we do. So this is gated on CD4 cells, and FOXP3 is in this direction, and CD25 is in this direction, and the regulatory T cells in here. And this was in a blog. It said, sometimes I wish that T-Rex rather than T-Regs regulated the immune system. Anyway, I just thought it was funny. Anyway, science jokes, I know, I'm sorry. Sorry, there's my disclosures. Okay, so I just want to just very briefly, every year I come and I sort of give an update. Genes are still coming fast and furious in this field. I was at the ESID meeting just a few months ago, and there were probably, I didn't tally them up, but there were probably 15 new genetic defects reported at the ESID meeting. It's on the order of about one per month in terms of a new genetic immunodeficiency. Things are moving along pretty quickly, and I think the big explosion in the last year, and I'll show you some examples of specifically related to this, is that within CVID now, several groups who are looking it's been estimated that we're now able to find the genetic cause in 25% of patients with CVID, which I think is a real breakthrough, because we've not really had, CVID's kind of been a black box. Largely, there were a few years ago, of course, there was a lot of excitement about TACI and some of these other things, but TACI really turned out to not be nearly as exciting as it was originally pitched to be. But several of these new genetic defects are actually, they're real. They actually really do cause disease. Interestingly, the majority of them are autosomal dominant, so that it only requires one mutation to get the phenotype. And so these, it sort of is the answer to all of these sporadic cases of CVID that we see where there's no family history and suddenly you get someone who's severely affected. And so it provides some examples of that. And so just as an update, again, in all of the different compartments, we're now at about 270 molecularly defined immunodeficiency disorders. And right now there's about 21 genes that have been associated with a phenotype of SCID. And it's been interesting as the data that came out this past year from the first 11 states doing newborn screening for SCID, there have been a few surprises in that. And among them are that the TREC analysis has picked up some kids with ataxia telangiectasia, for instance, who present with a SCID-like phenotype. That's been sort of a surprise. A disease that we didn't traditionally think of as necessarily having a SCID phenotype at birth. There have now been at least, there were 2 cases reported in the JAMA article. I'm aware of 2 more cases that have been identified by other groups. So it's one of those genes that we didn't traditionally think of as being a SCID gene that's now been added to this pool of SCID-associated genes that can show up in newborn screening. So I think that's been one new finding just over the past year to a year and a half as we've, with newborn screening and the availability of next-generation sequencing approaches, finding some old friends, I would say, that are expanded phenotypes as we're looking at these patients with SCID. Just in terms of concepts related to autoimmunity, some of you have seen this idea before when I talked about it. Which the first idea that garbage disposal in the immune system is really important. It's important to get rid of dead and dying cells. And the idea that if you don't get rid of these, what happens is that nucleic acid, acetylated proteins, various things that are known to activate innate immune pathways can hang around. And as they hang around, they drive production of type 1 interferons. And type 1 interferons, of course, have been associated with the generation of autoimmunity. And particularly, there's been a type 1 interferon signature that's quite well established in lupus. Type 1 interferons, of course, drive a lot of innate immune responses, particularly neutrophils and other innate dendritic cells and other innate immune responses, and are thought to play a role in the pathogenesis of certain vasculitides and other autoimmune disorders. And so we know that to get rid of these cells, there's really sort of multiple steps here. To get rid of these, opsonization by complement is important. And, of course, phagocytes are important for getting rid of those. And then, of course, as these hang around, there are innate response pathways in almost all of our cells that allow them to respond. They're there. These innate response pathways are there to help us respond to viruses and things like that. And so it turns out that defects in all of these can cause problems. So complement deficiency, as you know, is highly related to development of autoimmunity, and it's either lupus or a lupus-like glomerulonephritis. And this case was really a new case this year. There's been reports. There's been another report in the past which was unsuccessful, but this was exciting. This was a case report of a 10-year-old Pakistani male who presented with a 2 to 3 week history of headache, nausea, and mental status changes. Had a high titer ANA, or moderate titer ANA, but had high titer anti-Rho antibodies, positive anticardiolipin antibodies, anti-double-stranded DNA, and was noted to have mildly low C3, C4 at the time, was diagnosed with a CNS vasculopathy related to the mental status changes, and was thought to be probably related to lupus. This patient was treated with high-dose prednisone pulses and IV Cytoxan, sort of the usual approach that we take in patients who have lupus with CNS involvement. The patient had a facial rash and high titer, again, anti-Rho antibodies and anticardiolipin antibodies that just, despite this therapy, just did not seem to get much better. And so he was treated for he received sort of a usual course, 12-dose course of high-dose Cytoxan and methylpred, and then was transitioned to rituximab, sort of a usual approach to treating lupus. And then, as he was a little bit older, there was a 17-month-old brother who died of acute strep pneumonia meningitis, and that made the team that was caring for this patient say, that's odd, and went back and looked more carefully at the complement pathway, and this patient was found to be C1 deficient, C1q deficient, and this was, he had absent C1q. This was the picture of him. His facial rash, again, was very resistant to any of the therapies that he was given. Partly, again, because these therapies were not targeting the underlying problem, and he had ongoing problems, very difficult to control lupus. And so, because C1q is made both by the liver and by bone marrow macrophages, the thought was that bone marrow transplant may be of benefit in a patient with C1q deficiency. And of the complement deficiencies, C1 deficiencies, C1QRS, are the ones that are associated with the highest risk of severe autoimmunity, and they're the ones that we just about 6 months ago diagnosed a family actually with 2 young children who have C1 deficiency, and the problem, it's really kind of dreadful to sit down with them and tell them, if your kids don't die of infection, there's about a 95% chance they're going to get severe autoimmune disease, and they're going to run into problems. And this was really a nice case report. There had been a previous failed attempt at bone marrow transplant. This attempt, however, was successful. The patient underwent bone marrow transplant, and this is his facial rash now gone, post bone marrow transplant. And so this is, I think, a new, brings to light a new idea in terms of treating C1q deficiency. The point with this, however, the tricky point to the bone marrow transplant here is that with bone marrow transplant, sometimes in immunodeficient patients, we see development of split donor chimerism, what we call split donor chimerism after the transplant. If we don't, if patients are not aggressively conditioned, sometimes we don't get a myeloid graft that's stable. A lot of times what we'll see is that the patients will actually, their myeloid graft will, over time, go away, but they'll retain their lymphocyte graft. And for a patient with SCID, that's fine. That's all they need. They just need lymphocytes. With SCID or IPEX, where the T cells that are missing are the critical part, that's just fine. But in a case like this, where you really need bone marrow macrophages, you need to probably condition more aggressively so that you can get a stable myeloid graft in, because otherwise you won't cure the disease. And I think in reality that's probably what happened with the other failed attempt, is that they over time did not maintain a stable myeloid graft. And I think that's going to be the challenge with this disease. Again, with only very few patients reported in the literature doing this sort of prospectively. When do you do this in a family? If you identify a family with C1q deficiency, like we just did, 2 young children, both of them under the age of 3, do we tell them we want to take the kids to bone marrow transplant now, or do we wait until they develop lupus and then maybe control them with plasma infusions, as has been reported in a few case reports and a small case series? Control them, get them under control with plasma infusions, and then take them to bone marrow transplant. It's a real question, and I think we don't yet know, but it's exciting because it provides an opportunity potentially for a curative therapy in what has otherwise been a very difficult to treat disease. Now, many of the other complement components are made by the liver, and there has been discussion in the field, and there are anecdotal reports of the thought that a split liver transplant from either a related or unrelated donor could potentially be of benefit in patients with other complement deficiencies who develop autoimmunity. So I think that there are some evolving approaches to treating these disorders that I think have largely, we haven't thought about them a lot, I think, for a lot of years since they've been described. And these patients, they're rare, they're not very common. And so there's not been a lot of work in these fields, but the outcomes are absolutely abysmal in these patients who develop autoimmune disease, because treating the underlying disorder, the complement deficiency, can be quite difficult. You can give them plasma infusions, but that's a difficult way to live for a long time. So anyway, I think this is something new this year that is exciting and provides some new hope for these patients.
If you don't make C1q, do you make R and S? Are they separate?
Yeah. So yes, they are. So, but if you don't have C1q, R and S, of course, are part of that complex, and they don't do anything. So, at least they're not very efficient. So, sort of along the garbage disposal idea, again, many of you are aware that carriers of X-linked chronic granulomatous disease also develop discoid lupus. We think, again, it's a garbage disposal problem. They don't clear out probably pieces of dead and dying cells maybe quite as efficiently. And then this group of disorders have really emerged and are really expanding. These now all together are called interferonopathies. And these disorders, most of our cells have inside them innate components of the innate immune response that are in place to deal with nucleic acids. And the idea is that these nucleic acids would come, these are to deal with viral and maybe fungal, other intracellular pathogens that have nucleic acids. Of course, you've got toll-like receptors that deal with nucleic acids, that deal with sort of extracellular nucleic acids and endocytosed nucleic acids. These are nucleic acids that would be generated inside the cell. These pathways have been hypothesized to deal with viral nucleic acids, but also to deal with the bulk flow of, within our genomes, we've got there are a ton of retroviral elements that are endogenous retroviruses that have gotten themselves in there over the centuries or eons that humans have existed. And those, of course, are replicating and popping in and out probably of the genome. And people who study these pathways think that these innate response pathways are there to deal not only with viruses, but also with this bulk flow of retroviral elements that are endogenous to our genome. And so what happens typically is that the nucleic acids, whether it's double-stranded RNA or single-stranded or double-stranded DNA, are sensed by these pathways. And so some of these will ring a bell. RIG-I, MDA5, this is also known as IFIH1, and TREX1. The nucleic acids are either degraded, such as by the TREX1 complex, or they're sensed, and the sensing of these ultimately leads to the activation of type 1 interferon generation, and type 1 interferons then, of course, drive responses. Part of the response is a cell intrinsic response, so that it shuts down protein translation and other things within the cell, but it also then attracts inflammatory cells, so that if you have a virus, these are some of the danger signals that are sent out to attract inflammatory cells to the site to deal with this virus. Now, we've known for a few years about the association of DNase 1 defects with in lupus. TREX1 defects have been identified in a particular type of familial, what's called Chilblain's lupus and Aicardi-Goutieres Syndrome. But really, the new thing over the last year have been, and these are loss of function mutations. So these are mutations where if these are lost, again, these are related in degrading the DNA. So you can imagine if these are lost, the DNA accumulates, drives type 1 interferons. The two, really the new players in this pathway are these gain of function mutations in these DNA and RNA sensing pathways. So these are actually autosomal dominant. So it only takes one mutation to cause the disease. And what they do is they make these molecules more sensitive to be triggered. So MDA5 or IFIH1, for instance, these activating mutations make it so that just a little binding of nucleic acid sets off this pathway. And so as you can imagine, if it doesn't take much to get the pathway going, these cells are going to be making type 1 interferons, a lot of them, kind of all the time. And similarly in STING. And so over the last year, we've come to understand more about these. Again, these patients with defects in this pathway can develop Aicardi-Goutieres Syndrome. For those of you who maybe don't do pediatrics, just to introduce you to this disease, this is a progressive encephalopathy that we see in little kids typically. They're born and over time they develop calcifications in characteristic areas of the brain. This has been confused in some cases with cerebral palsy and other with congenital infections with toxoplasma and other congenital infectious organisms have calcifications in a similar place. So these patients often get worked up for those sorts of disorders. But over time, this is a progressive defect because, again, it's inflammation that's being driven by production of these type 1 interferons, and it's progressive, and these patients have poor outcomes. They may also have elevated liver enzymes and fevers, depending on how aggressive this inflammation is. But in addition to Aicardi-Goutieres syndrome, these type 1 interferons can also drive lupus. And so this has been described in a couple of cases. So this is the IFIH1 defect. This one was the one that was described. This article came out first in Immunity and was actually a mouse model. And these are deposition, sort of a classic lupus look in the glomeruli, like a lupus-like glomerulonephritis with deposition of complement and antibodies. And shortly thereafter, the human patients were described with IFIH1 activating mutations. Most of them had Aicardi-Goutieres, but some of them had lupus. And again, this is what's called a type 1 interferon score. And this type 1 interferon score, this is 6 genes that are driven by type 1 interferon that you can actually just measure in bulk blood by quantitative PCR, and is actually a really nice tool to identify these patients among, for instance, patients with early onset lupus. And so patients who have high type 1 interferon mediated genes have of course, all of these genes are that are driven by type 1 interferons are very elevated compared to normal individuals. And if this persists over time, that's a suggestion that in fact the patient may have one of these disorders. And what's come to light, so this is the other okay, where'd it go? I lost it. There was one in here. No, I lost it. Oh, accidentally deleted one slide. So the other defect in this is STING. I highlighted STING. So STING was, if you look here in this pathway, STING is another molecule in this pathway. And recently there have been activating STING mutations that have been described. They drive a vasculopathy it's a vasculitis. Some of the patients look like lupus, and this was a patient that's not yet published, but turned out to have a STING mutation. So this was a 48-year-old Caucasian male who developed lupus 6 years ago, including rash, arthritis, glomerulonephritis, and antiphospholipid antibodies. And the symptoms were moderately controlled with corticosteroids and rituximab. And then he had a daughter, and his 3-year-old daughter has now developed a photosensitive rash on her face and hands, and she's also been complaining of her legs hurting, particularly her knees. She's got an ANA positive, a positive double-stranded DNA, and because of the association of lupus in a young child, the father had lupus. They went looking for these defects, and in fact found an activating STING mutation in these patients. And so these patients with these these activating defects in these type 1 interferon pathways are floating around out there. They're probably among the patients followed in the rheumatology clinic. These are actually immunodeficiencies, or I should say immune dysregulation disorders, really is what they are, that present with autoimmunity, and they're defects in the production of type 1 interferons. And so I think the point that I'd just like to leave you with is the idea that type 1 interferons drive vasculitic diseases. So if you just have a lot of type 1 interferons, and we know that in patients who've been treated with type 1 interferons that there is a risk for developing some autoimmunity in those patients. And it's been proposed that in these patients, because type 1 interferons are thought to be a driver, it's been proposed that the use of JAK inhibitors might actually be beneficial in these patients. And again, you would, in rheumatology, JAK inhibitors are not among the molecules that I think about for treating a patient with lupus, and particularly not severe lupus. And so again, the critical point is to go looking. If you've got patients who have symptoms or a familial pattern of inheritance, to go looking for these disorders, because it may actually point the way to a therapy that you would not otherwise think about or is not traditional in these diseases. And so I think that this is one of the things that has come out, really been strengthened much more in the past year, the relationship between autoimmunity and immune deficiency or immune dysregulation with known molecular defects. My prediction is that there will be several more defects that lead to production of type 1 interferons that will be produced. And this is going to be an ever-growing subset of disorders that have now been called, as a group, the interferonopathies. Okay? So,
is there anything about this new information and the fact that women get these diseases much more than men? Is there any hormonal?
No, not yet. It's a great question, and I think there's no data yet as to what how that might play out. So, okay. Let's talk about some regulatory T cell defects, and of course I'll start by reminding you that in terms of tolerance in the T cell system, of course there's all of the tolerance mechanisms that take place that are central tolerance, that take place at the level of the thymus, and you've got positive selection, negative selection of autoreactive T cells to get rid of cells that shouldn't be causing. You don't want them to cause autoimmunity, so you get rid of them in the thymus. Those are all central tolerance mechanisms. And in comparison, peripheral tolerance is mediated, we think, largely by regulatory T cells. And there's the sort of the classic CD4 positive, CD25 positive, FOXP3 positive regulatory T cell. And there are other regulatory mechanisms that are at play in the periphery. Regulatory T cells are generated in the thymus. This is from Sasha Rudensky, and interestingly, we still actually don't know all of the mechanisms that lead to the generation of a regulatory T cell. We know that their T cell receptors are a little bit autoimmune. They're not strongly enough autoimmune to get them deleted and negatively selected, but they're a little bit autoimmune. They've got a higher self-reactivity than the other population of effector T cells. So it's related to the T cell receptor response. When the T cell receptor is a little stronger, but not too strong, they get turned into regulatory T cells. And blue is the thymic cortex. Red are the thymic medullary epithelial cells, and green are the regulatory T cells. And what you really note in this slide is that the regulatory T cells are pretty much only in the thymic medulla. And we know, in fact, that that's where they get generated. T cells are just becoming single positive CD4, CD8 as they transit then from the thymic cortex into the thymic medulla. And it's really there in the thymic medulla where the Tregs, the classical Tregs, get generated. And there are a number of mechanisms that we know are important, but there are a number of mechanisms that we still don't know, despite really this being studied intensely for the last couple of decades. So I think that there's still a lot of unanswered questions here, but the regulatory T cells are generated here, but then they exert their effect throughout the body in the periphery. And regulatory T cells have a lot of important characteristics. One of them, of course, they express FOXP3, which is the transcription factor that's required for their development and function. They express other important molecules. So one of the characteristic things is they express high levels of CD25, which is the alpha chain of the IL-2 receptor. So regulatory T cells are very responsive to IL-2. In fact, they require ongoing stimulation by IL-2 to keep their FOXP3 levels up so that they can stay a regulatory T cell. So they require ongoing IL-2 stimulation to do that, and that's why they express CD25 all the time. It's the high affinity IL-2 receptor, and that allows them to sense just minute amounts of IL-2. They also express co-stimulatory molecules like CTLA-4 and GITR and some of these others that allow them to in cell-cell interactions to regulate cells, and we'll come back and talk particularly about CTLA-4. And then they have adhesion molecules that allow them to get around in the body. They also secrete cytokines, and some of their effects are mediated through direct cell-cell interactions, and some of their effects are mediated through the production of important cytokines, things like IL-10 or TGF-beta that shut down bystander cells and are able to mediate immune suppression in those ways. And so I just wanted to talk through some things related to these, a couple of new findings related to these that I think are important. And so let me go through those. This is IPEX, again, FOXP3 deficiency. I've shown this before. These patients are very sick early on. They develop enteropathy, type 1 diabetes, thyroid disease, and dermatitis. I think the thing really that we've come to realize, of course, these patients have no regulatory T cells. The thing that we've come to realize, one of the things over the past couple of years, is that in the absence of regulatory T cells begin to function in utero. And this is data from a patient that we performed an autopsy on here. This patient died at about 36 hours of age. So this was a child who was a prenatal diagnosis that we had actually transplanted his older brother. We knew that he was affected, and in fact we had chosen a bone marrow donor for him prior to him being delivered, with the idea that we would transplant him immediately after delivery and prevent any autoimmunity. His brother had a severe course, and we anticipated that this patient would have a severe course as well. Near the end of pregnancy, he was noted to have small lungs, and there was a concern that the patient had a diaphragmatic hernia. And so he was transferred the mom was transferred from her home from San Diego up here so that we could deliver the child here and potentially move him immediately to transplant. So she was moved here, monitored for a couple of weeks, and it became more apparent that the lungs were quite small in this child, more and more concern for a diaphragmatic hernia. The child was born, did not have a diaphragmatic hernia, but the chest was quite small, and the child could not be ventilated. And it's something that we've come to realize now in a small subset of IPEX patients. We've now got 4 patients who had fairly significant pulmonary hyperplasia. This child had profound pulmonary hyperplasia. Lungs were about 1/3 the size that they should have been for his gestational age. Interestingly, completely normally developed. So we have no idea why they were so small. He didn't have a diaphragmatic hernia, didn't have any other of the classic reasons that we think about for small lungs in babies. But what was interesting was that at autopsy, it was evident that the autoimmunity had already begun in earnest in utero. And it's interesting because you think about the uterine environment. traditionally in immunology, we think about the uterine environment as being very immunosuppressive. It allows mom to carry this haploidentical, half-matched, tumor-like thing in her belly for 9 months without rejecting it, right? And so there's very significant immune tolerance mechanisms at play in pregnancy. And you'd think that those might be enough to prevent the autoimmunity, and in fact, they're not. The baby's regulatory T cells begin to function in utero and are required to prevent autoimmunity. So let me show you. So here is the H&E stain of the pancreas. So here's a normal control infant who died at a similar age of an unrelated problem, and again looks pristine. And here is the patient, and you'll note there's a lot of inflammatory infiltrates here. You can see the darker, sort of just general darker staining of inflammatory cells. And when those are stained for T cells, CD3, CD8, CD4, CD8, and CD20, for B cells, they robust inflammation in the pancreas. And this child is 36 hours old at this point, and it's already well underway. And the interesting thing for us was to note that, in fact, the inflammation in the patient, if you look at these, so these are the T cells, and you note here are sort of these little light areas, and if you look over here, those are actually the B cells. So this is like a follicular inflammation in the pancreas. And what was interesting to us is that the inflammation in this child's pancreas was almost exclusively in the exocrine pancreas. Almost did not touch the endocrine pancreas, which is interesting because these patients get type 1 diabetes. And so it's actually made us rethink, and we've had several patients who've had exocrine pancreatic dysfunction, and it's actually made us rethink the type 1 diabetes in this disorder, that in fact maybe the type 1 diabetes is actually collateral damage from an aggressive exocrine pancreas attack that leads to then collateral damage causing type 1 diabetes subsequently. And as I'll show you in a disorder, I'm going to talk about CTLA-4 haploinsufficiency in a moment. Interestingly, in the CTLA-4 deficient mice, they have a very similar phenotype to the mice that lack FOXP3. Very early onset severe autoimmunity, lots of lymphocytic infiltrates in multiple organs, including the pancreas. And in those mice, they also have exocrine pancreatitis. And so it may be that, in fact, the mechanism of disease here is different than we thought, and we're doing some work to look into that. Now, just as a reminder, these patients, the way that we treat these patients is with aggressive supportive care and then and then T-cell directed immunosuppression followed by bone marrow transplant. We've not found any of these patients who've lived beyond their 20s and 30s. There are some milder cases that do live into their 20s and 30s. We just transplanted a 24-year-old patient here who was in terrible shape, and so we still advocate early bone marrow transplant for this disease. Now I want to get back to the regulatory T cells and highlight a disorder that's really been identified and we've learned much more about in the past year, and that is CTLA-4 haploinsufficiency. Now it turns out in mice, again, mice are not equal to humans, as we know, but this is a particular difference. In mice, if you make them heterozygous if you make a CTLA-4 knockout mice and make a heterozygote, so they've got CTLA-4 expressed on one allele, not expressed on the other, they're fine. They have no clinical phenotype. You have to lose all CTLA-4 to see a phenotype. And as I mentioned, the phenotype of CTLA-4 deficient mice is that they develop an autoimmune syndrome similar to what we see in the FOXP3 deficient mice. Very aggressive autoimmunity. They die within a few weeks of life. And they have really profound lymphocytic infiltrates in lung, in heart, and in other organs. And we have been looking actually for several years for CTLA-4 deficiency in our IPEX-like cohort, the patients who have an IPEX phenotype but don't have FOXP3 mutations. And we hadn't found any. And interestingly, over the last year, there have been 2 papers published just recently identifying CTLA-4 haploinsufficiency in humans. Now, this is haploinsufficiency. So unlike the mice, in humans, if you knock out one allele of CTLA-4, it's actually enough to develop disease. And I want to show you the data from these 2 papers because I think it's really interesting. And it turns out that this is, within the CVID population, is moderately or modestly common. There's a fair number of these patients in the CVID population. Again, it's not, but it probably accounts for maybe 5% of the CVID patients in cohorts that have been studied. So in Bodo Grimbacher's cohort, for instance, that he studied, it was around 5% of patients had CTLA-4 haploinsufficiency. And the phenotype is really interesting because it's that type of CVID that has all of the autoimmunity associated with it. So it's the bad type of CVID, not the ones who just get infections. And so this was the NIH paper. The patients, these were their pedigrees. Again, because it only requires a mutation in one allele, it's autosomal dominant. So you don't need to have a family history. It can occur sporadically. And so this was their cohort showing the affected patients in red, and showing here that these patients, again, because CTLA-4 is an important co-stimulatory molecule on regulatory T cells, they looked at the regulatory T cells, and in this paper, they showed that their regulatory T cells, which would be here in this quadrant, here's a normal individual, are low. Now, this was not consistent in the other paper that I'll show you in a minute. Bodo Grimbacher's paper did not see as big a difference using the way that they looked at regulatory T cells. But there's clearly a regulatory T cell problem in these patients. Now, what does CTLA-4 do? CTLA-4 is a co-stimulatory molecule. Remember that when T cells get activated, they require 2 signals. They require a signal through the T cell receptor, and then typically CD28 is another co-stimulatory molecule that interacts with the B7 molecules on dendritic cells and other things to provide that second signal that T cells need to get stimulated. And CTLA-4 also interacts with that B7 family of molecules, but it has the opposite. It downregulates the B7. So what it'll cause to happen when CTLA-4 interacts with those B7, those co-stimulatory molecules on dendritic cells, it will actually cause B7 to be pulled off the surface of the dendritic cell, so it can't co-stimulate a T cell. So that's one of the things that CTLA-4 does. It downregulates the ability of dendritic cells to co-stimulate T cell activation. And so that's one of the ways that CTLA-4 works. And it's also been shown that CTLA-4 makes it so that T cells, when it's expressed, the cells don't traffic into sites of inflammation as well. So CTLA-4 is important in a couple of steps in regulating immunity. So this is the NIH paper. This is the paper from Bodo Grimbacher's group. These are his cohorts. And the important thing that I want to highlight here is that it turns out that as they looked in all of the patients in the cohort, it turned out that there were patients who had the mutation who did not have symptoms. So it's not completely penetrant. Okay? So that was one important thing learned from these large cohorts, is that in fact these gray patients had the mutation in CTLA-4, but they did not have symptoms either of significant autoimmunity or of immune deficiency, whereas the patients in black had the full phenotype of either CVID or autoimmunity. And so these were the patients that Bodo studied. Okay? Now, the important thing we think in recognizing these patients is that these are the CVID patients who get these lymphocytic infiltrates in lung, gut, and interestingly in brain. And I would say that if you have a CVID patient who develops brain lesions, this is the place you look, first and foremost. I mean, it's unbelievable how many of these patients have developed brain lesions with lymphocytic infiltrates in the brain. And I'll show you sort of the breakdown of clinical symptoms, but this is gut here, as you can see. And look at these things are just loaded with lymphocytes. These are just T-cell infiltrates, just uncontrolled T-cell infiltrates. And in the lung, this is sort of that nodular lymphoid hyperplasia, that GLILD picture that you see in some patients with CVID. And I'll show you some more pictures in a minute. These are just packed with lymphocytes. And the same thing in the brain. Here's the brain of a patient. Look at this thing. It's a massive lesion. You'd almost think it was a brain tumor, and in fact it wasn't. It was just lymphocytes in the brain. Really, really impressive. Here's some from the other paper. Again, gut with lots of T cells in the gut. This is lung. Again, this is that follicular bronchiolitis that we see often in CVID patients when we biopsy their lungs. We'll see the B cell zone, the T cell zone. Pretty classic for GLILD, the sort of stuff we see in patients. Again, look at these. These just look like the GLILD lung that we see in our CVID patients. Nodular lung. They have the nodular lymphoid hyperplasia in the gut, lung again, and brain. And again, here's another brain lesion that you might think is a tumor. And in fact, it's not a tumor. It's not a lymphoma. It's just out of control aggregated lymphocytes.
Are most of these brain lesions symptomatic?
Yes. Well, no. I should say no. They're not. So when they went, so they looked at a few so we don't know yet. When they did look, Bodo looked at a few asymptomatic patients, and they did see some little ditzels in some of those patients.
Should we be getting brain scans on our patients? Because we don't.
Boy, it's a great question. I think if you've got a patient who's got really impressive, I mean, I don't know that it's going to necessarily change your management, because I think what you should get, if you've got a patient who's got lungs that look like that, you definitely should get a lung biopsy. And probably again, we've been treating patients who got lungs that look like that. We've been treating them with rituximab and azathioprine, sort of following the approach that Jack Brutus has sort of advocated. And it works. And my suspicion is it will probably work in these. It may not be, because you're not really targeting the T cells, of course, with rituximab, but the azathioprine would help. I think, unless they're symptomatic, I'm not sure that it's necessarily worth the expense. I mean, it might, again, target you, but we're going to be sequencing all these patients anyway, I think.
They started pushing the transplant for the CTLA-4.
So that's an excellent question, and in fact, no. So people have not, and it would make sense that this would be a cohort that they would be fixable by transplant. Really, the therapy that has been most effective so far that's been reported, well, it's been reported a little bit in the NIH paper, was they are using sirolimus in this patient population. Sirolimus, again, remember, acts on the mTOR pathway. That's mTOR signaling, of course, is important for integrating all of these signals that come into the cell, come into T cells about nutrition and cell size. It plays a really critical role in regulating cell growth and metabolism. And in T cells, it plays a central role. The cool thing about mTOR inhibition, rapamycin, and all of the other related molecules, sirolimus, everolimus, there's a whole family of these now, is that they clamp down on the mTOR pathway, and because T cells, effector T cells, require this pathway to get activated and grow and divide, when you clamp down on that, they can't do that, and so it clamps down their growth, and it's very effective in that manner. The cool thing about rapamycin in a disorder like this is that regulatory T cells are not dependent on the mTOR pathway for their growth. They're resistant to mTOR inhibition. So it allows you, it's a drug that allows you to clamp down on effector T cells while allowing regulatory T cells to function. So it's different than cyclosporin or FK506, because cyclosporin and FK506 clamp down on both and do not promote tolerance. Sirolimus, or rapamycin, promotes tolerance because it clamps down on effectors and allows Tregs to expand and do their thing. Okay, so in disorders like this, it potentially has sort of a really nice added benefit. So these patients, again, So diarrhea and enteropathy, again, sort of thinking about those IPEX-like kind of features. It's not as severe as the IPEX patients, but they definitely have, these patients have the type of diarrhea and enteropathy that we see in CVID patients, which is a lymphoid nodular hyperplasia when they're biopsied. And it's very problematic for many of these patients. They have hypogammaglobulinemia in almost all of them, so they have a CVID-like phenotype. And it's not clear why this happens or what the mechanism of this is. GLILD, granulomatous lymphocytic interstitial lung disease. They have, of course, respiratory infections related to their hypogammaglobulinemia. They have organ infiltration, again, as I've shown you, just these profound lymphocytic infiltrates in organs. And then, of course, they have lots of other autoimmunity, as you can see here, psoriasis, lymphadenopathy, et cetera, et cetera, et cetera. So lots of problems. So this disorder, I think, as I mentioned, in Bodo Grimbacher's cohort, in what he talked about at the ESID meeting is that it's about 5% of his CVID patients have this. It's a small gene. It's only 4 exons. It's easy to sequence. We're ramping up to offer it clinically. We're not yet offering it clinically, but if you've got patients that you're interested in, we'll be happy to sequence them for you. And I think it plays a major role because if you can find this defect, again, you might think about taking the patients to transplant earlier. I think that transplant would be perfect in this disease. It would cure this disorder because it cures the mites. So bone marrow transplant in this disease would be excellent. And so I think that again, these are patients that you can really make a big difference for. And knowing the defect early allows you, I think, to intervene more aggressively earlier in the disease because you've got an answer. Okay, so CTLA-4 haploinsufficiency, I think, is a new defect this year that I think is really important in this population of CVID with a heavy autoimmune overlap. Okay, so I just wanted to just remind you that I'm going to step down here that there are published at the ESID meeting. They're not yet in press yet. There was another defect associated with CVID. So I've shown you this before. I showed you this last year that there are now 2 different disorders in the 2 different subunits of PI3 kinase. So there's a P110 subunit and a P85 subunit, and now defects have been identified in both of those subunits. These have a CVID phenotype, also with granulomatous lymphocytic sort of immune dysregulation. You'll note that all of these lie downstream of the B cell receptor. Of course, patients with autosomal dominant defects in what's called NF-kappa B2 in the non-canonical NF-kappa B pathway, and the other new defect that was published or that was presented at the ESID meeting, which is not yet in press, and accounts for actually again a fair number of patients in Bodo's cohort, is defects in NF-kappa B1 or NF-kappa B P50, and these patients it's a haploinsufficiency of this subunit of NF-kappa B. These should be coming out in press, I would think, pretty soon. And again, these patients are in the CVID cohort and have some interesting phenotypic features. So just to again highlight a couple of new defects that have been identified just within the last year or so. Both subunits of these have now been published. These have not yet been published, and there will be more patients with this defect. That another one is now in the press, and there are more that are coming. So we're learning more about these pathways and the development of CVID. And you might predict if these lie downstream of the B cell receptor, that in fact, if you don't get good B cell receptor signaling, it makes sense that you're not going to get great B cell development and function in the long run. Yes?
If you look at adults who present with CVID at an older age, do they have these same mutations?
Yes.
Just from lack of function?
Some of them do, yes. Mm-hmm. Yeah, because there's variable penetrance for some of these. So they found them in older patients. Some of those older patients, when you go back, they'd had symptoms, since they were maybe teenagers or younger kids. But, that's not that uncommon in our CVID population. Especially the study that the Immunodeficiency Foundation did, the average delay to diagnosis was 8 years, that patients could sort of go back and say, my symptoms started 8 years ago. 25% of patients had a 20-year delay to diagnosis. So yes, they are finding these disorders in adults. And so it's still worth thinking about. Now, are these going to be the defects in a 73-year-old who presents with new onset symptoms in the last 3 years, say? Probably not. But if you've got a 35-year-old or a 40-year-old who's had symptoms for 8 or 10 years, when you sort of track back, absolutely, I think it could be in those patients.
If these are genetic defects, why do you get that kind of a history that they're in their 30s and they presented 8 years ago? Why didn't they present at infancy?
It's a great question, and I think we don't understand enough about them yet. Again, these are heterozygous. So for instance, in CTLA-4 and in this these NFKB1 defects, they're haploinsufficiency. So the patients still have half of the normal amount. So that may get them by for a while until maybe they run into some infectious challenge or some other environmental challenge that sort of gets the immune system revved up and they can't handle it, and that then sets off this process. And so we don't really know. Or are there other modifier mutations? Interestingly, I was talking to Jordan Orange a couple of months ago. We were at a meeting and chatting, and they've, with their genomics center at Baylor, they've been sort of plowing through a lot of their undefined immunodeficiency patients. And they've actually found that 1 in 20 patients where they find a genetic defect, 1 in 20 have 2 immunodeficiencies. They've got genetic defects in 2 different immunodeficiency genes, that either of which could cause disease, but their phenotype is some sort of mishmash of both of those defects. And so it's not, in medical school we were all taught, find the unifying diagnosis. Well, it turns out there may be 2 unifying diagnoses, actually, and they may both be genetic. And so we don't yet know. I'm sure there's probably some role for modifier genes there as well. So again, there's a lot more to learn in these disorders. It was interesting at the ESID meeting, it was just rapid fire, new gene, gene. And when you pin them down about the function and the mechanistic studies, almost none of that has been done for most of these. It's sort of the gold rush for genes right now, and the mechanistic studies. At some point we're going to saturate the genes. We're going to find all of the immunodeficiency genes, and then people are going to have to go back and do the hard work of actually doing the mechanistic studies and understanding what these things actually do. So I think that's really, that's where there's a lot of work left to do. Thank heavens, keep us in business for a while. So anyway, I'll stop there. I think there's lots of new interesting defects. I think this area, this idea of immune dysregulation and primary defects that lead to immune dysregulation has really become of great interest in the immunodeficiency field. And my guess is that within another 10 years, we're going to be looking at 200 genes that the rheumatologists need to think about. And this is going to bridge over into rheumatology in a big way, I feel, in the next 10 years. It's already beginning to do that a little bit in the pediatric rheumatology population, and my guess is on the adult side it's going to happen as well. So anyway, I'd be happy to take any other questions and answer anything else you might have.
Sure.
All right, thanks, guys.
I had a somewhat facetious question on one of the articles that we had all the authors listed at the top.
Yeah.
I was wondering how many more authors there are than patients.
Oh. Yeah. Team science. Yeah, right. That's the mantra these days, right? Big science, isn't that it?
Yeah. The chase of
idea of bone marrow transplant.
Highbrow today.
Until Matt figures out how to do it, I'll be into a bone marrow transplant. That'll be his goal. So Matt, what do you know about the CAR T cells? Do you know that? You doing any of that with children?
The CAR T cells? I know what it is.
One of my, I was skiing with Phil Bensinger at the Hutch. He was talking about these people who interact with cancer. And they grow up these CAR T cells and they go into remission like immediately.
So, we talk about it. I don't think any
Actually, about the response of Tregs protecting your shorts. I think people think that it's mainly the
The Swedish is evidently they're banking on this being cancer of the future, directed gene therapy. You know, they're banking on CAR T cells.
I mean, it's a big overlap.
I think so.
Follow on. A lot of the immunologists are doing is trying to go.
That could be really interesting.